Welcome to this explainer on sub-agglutinating antibody titers. Look, if you've ever stared at a row of test tubes or a microtiter plate in the lab and just felt a bit, well, overwhelmed by all the math and the terminology, you are definitely in the right place today. We're going to completely demystify agglutination, calculate some titers, and tackle those tricky sub-agglutinating ranges, all without getting bogged down in messy equations. Let's get you feeling totally confident for the lab bench and, of course, your upcoming exams. Here's our roadmap for today. We're covering 1. The basics of agglutination, 2. Serial dilution and endpoint, 3. Calculating antibody titers, 4. The sub-agglutinating range and zones, 5. Standardization and controls, and finally, veterinary serology in action. All right, part 1. The basics of agglutination. So what exactly is agglutination? Fundamentally, it's an immune reaction. It happens when specific antibodies bind to particulate antigens, think whole bacterial cells or sheep red blood cells, and cross-link them into visible aggregates or clumps. Honestly, it's like throwing a microscopic net over a bunch of floating balloons, tying them all together until they form a large, heavy mass that you can actually see with your naked eye. But remember, this macro-level visual clumping only happens when the right antibody meets its specific antigen target. Now, how do we actually make this happen at the lab bench? Well, it's a beautifully logical four-step sequence. First, you prepare your antiserum. Second, you perform a serial dilution to progressively drop the concentration of those antibodies. Third, you add a carefully standardized antigen to every single tube. And finally, you observe where the agglutination stops, the endpoint. Moving right along to part 2, serial dilution and endpoint. This brings us to a massively important concept, one you will absolutely see on your exams, the antibody titer. By strict definition, a titer is the reciprocal of the highest dilution of serum that produces a defined positive reaction under standardized assay conditions. The absolute most crucial thing to grasp here is that a titer is an assay-dependent measure of detectable activity. It is not an absolute concentration. So it doesn't tell you exactly how many milligrams of antibody are in your tube. Rather, it tells you how far you can dilute that serum and still see that visible clumping. Okay, part 3. Calculating antibody titers. Let's do some math. Picture a simulated row of test tubes. On the left here, we're doing a two-fold serial dilution, starting at 1:2 and going all the way down to 1:64. Every step down exactly halves the antibody concentration. Over on the right, you'll see our visual reaction strength. See how at 1:2 and 1:4 we have a really strong 3+ reaction? At 1:8, it drops to a 2+. By 1:16, it's just a single plus. Then at 1:32, we get a plus-minus. That's a super weak borderline reaction. And finally, by 1:64, the reaction is totally negative. You can literally trace how the visible cross-linking decreases right along with the antibody concentration. All right, I want you to pause and actually think about this for a second. Based on the laboratory's strict definition that only a clear single plus is considered a positive reaction, calculate the antibody titer in your head right now. Find the highest positive dilution, and remember that definition of a titer we just talked about. Got it? If you said 16, you hit the nail on the head. Here is exactly why. Looking back at the sequence, the highest dilution that still gave a clear, solid plus reaction was 1:16. Since a titer is the reciprocal of that highest positive dilution, you just flip the fraction. The reciprocal of 1/16 is 16. It really is that straightforward. Let's dive into part 4, sub-agglutinating range and zones. Now, you absolutely must keep these two concepts separate in your head. First, the titer, which we just nailed down as the highest dilution giving a positive reaction, like that 1:16 tube. Second, the sub-agglutinating dilution. This is the dilution where antibody is definitely still present, but the reaction is just too weak to meet the lab's official visual cutoff. Think back to that plus-minus result we saw at the 1:32 dilution. That is your sub-agglutinating dilution. It's not completely negative, sure, but it just doesn't cross the threshold to be called positive. But here's the catch. Antibody concentration isn't the only player here. The proportion of antigen to antibody is absolutely critical. Let's break down 3 conceptual zones. The zone of equivalence is pretty self-explanatory. It's that sweet spot, the favorable proportions where extensive lattice formation gives you the strongest visible clumping. But then look at the prozone. This is wild and totally counterintuitive. If you have excess antibody, it actually prevents optimal cross-linking. You end up with a weak or even falsely negative reaction. Having too much antibody can literally ruin your visible clump. On the flip side, the postzone happens when you have excess antigen, which also ruins the cross-linking and gives a weak reaction.
Section 5, standardization and controls. This is how we keep things accurate. So how do we prove our results are real? We run strict mandatory controls. 3 major ones you need to memorize. First up, a positive control. You use a known antiserum to prove your assay can actually detect antibodies in the first place. Second, a negative control, using negative serum to prove you aren't getting weird, nonspecific reactions. And third, this one's huge, an antigen control. This is just the antigen all by itself. Why do we need this? Well, imagine if every single test tube on your bench suddenly gets clumpy. You absolutely need this control to prove those red blood cells or bacteria aren't just spontaneously aggregating on their own because of some bad reagents. When things do go sideways in the lab, these are almost always the culprits. These are the most common technical errors that will completely invalidate a reported titer. Messing up your serial dilution pipetting? Yeah, that'll ruin every single subsequent tube. Using unstandardized antigen concentrations? That's going to push you straight into that prozone or postzone we just covered. And finally, if you misread those weak, sub-agglutinating reactions as positive, you are going to falsely elevate your reported titer. Section 6, Veterinary Serology in Action. Let's tie it all together. Look, antibody titration isn't just a hurdle for your immunology class. It's a vital tool in clinical practice. We use it all the time for the serodiagnosis of pathogens to figure out if an animal has been exposed. We also use it to monitor herd antibody responses after a vaccination. To put that into perspective, say you take a blood sample before vaccinating a calf and you see a baseline titer of 8. You test again a few weeks later and bam, the post-vaccination titer is 128. That massive jump visually proves a highly robust secondary immune response. You literally just watch the vaccine-induced humoral immunity. So here is the ultimate master workflow you need to lock into your mental model for exam day. You pull the antiserum, perform the serial dilution, add the standardized antigen, observe the agglutination, identify the endpoint, calculate your titer, recognize the sub-agglutinating range, and then apply it to veterinary medicine. Let's wrap this up with your high-yield take-home points. Number 1, your titer is always the reciprocal of the highest positive dilution. Number 2, sub-agglutinating reactions are the ones that fall just below the defined visual endpoint. So not perfectly negative, but not positive either. And number 3, controls are non-negotiable. They are mandatory to rule out technical errors or that sneaky prozone effect, where too much antibody masks a positive result. Before we go, I want to leave you with one final thought. We just said that a titer jumping from 8 to 128 shows an incredible immune response, right? But if a vaccinated animal shows a massive increase in antibody titer on this in vitro lab test, does that automatically guarantee complete clinical in vivo protection against the disease? Well, not necessarily. A high titer tells us a lot about humoral immunity, but what about the wildly complex world of cellular immunity? It's a fascinating limitation of these tests, and exactly what you should be curious to explore next. Thanks for hanging out with me for this explainer, and happy studying.